Ooh, okay. I think this is rolling. I'm hoping that it's working. Guess please let me know in the chat if anything isn't working. But hi everyone, I'm Henri. Thank you for coming here. I'm from the Student Tutors, and today I was going to run through some questions for GCSE biology, looking at some past paper questions specifically on enzymes and the human digestion to do with sort of enzymes that are involved in that process. And the point of this quick little session that we've got here is to basically get you more comfortable with exam technique and to make you more comfortable in being able to identify what examiners are looking for when they ask particular questions and how to basically get the most marks that you can. So, without further ado, if anyone has any questions at any point, please let me know. If I can answer them there and then, I will do, but there'll be like 5 or 10 minutes in the end for some questions. What I'll do is I'll probably like read out the question, give you a few seconds or a minute, depending how long the question is, to sort of think about what the answer could be. I'll go through the answer, I'll go through my thought process behind it, and we'll see what we can get up to. So before we start, I'm just gonna pop a little message in the chat. Right, so without further ado, let's make a start. So here we have our first question, and so it reads that amylase is an enzyme that breaks down starch. Okay, so we've got given an enzyme here and its function. So amylase is a polymer of small molecules. Name the type of smaller molecule. So what is amylase made from? Now, some of you might be inclined to think to say glucose because amylase is very close to the word amylose. But amylose is in fact the same thing as starch, so it's a polysaccharide, in which case, yeah, it would be made out of glucose, but this is amylase. We've been told it's an enzyme. We know enzymes are proteins, and I know that proteins are made out of amino acids. So my answer for this one is going to be amino acids. Yeah. And now the next bit is going to be name the 3 parts of the human digestive system that produce amylase. So the 3 parts of the human digestive system, that produce amylase. Now for this one, you might remember the story of how amylose, as in starch, is digested. And you might recall that the first place of starch digestion happens within the mouth. So that could be one of your answers. And then you know that the rest of it usually happens after the stomach. So I'm going to first tackle the mouth. And you could be inclined to write just mouth, but it's asking you for the parts of the digestive system that produce this enzyme, and that is in your mouth. That's where the digestion happens. So the specific organs, in a way, that produce this enzyme in the mouth are salivary glands. So you can start there. And that's going to be our first place. And now the second place, as we said, after the stomach, is going to be the pancreas. So the pancreas releases loads of different enzymes into your small intestine after food travels into the stomach. And one of these enzymes is this amylase. So that will be our next answer. And last but not least is the small intestine. So small and small intestine is where these enzymes are basically in your membrane of your intestines, so they're not free floating like they are in the rest of your digestive tract. So our next question is explain how amylase breaks down starch. So here we've got to pay attention to the word explain. So it doesn't want us to just describe what we saw in terms of saying why the sun is just bright. It wants us to explain why. So the sun is bright because it's burning loads of gas, for example. So here it also then tells us to answer in the terms of the lock and key hypothesis. So the lock and key hypothesis is something you should be familiar with, but if not, we will go through it here. But that is something we need to reference at least somewhere here. So hopefully you've given the answer some thought. The first place that I'm going to start with is we've got amylase breaking down starch. So amylase is going to be our enzyme. So I'm just going to write a little E over this. And starch is going to be our substrate. So I'm going to write a little S over this, just so I remember which one's which. And what we know is that substrates bind to an active site. Now you need to be careful to not just say bind to the enzyme. You need to say 100% that it binds to the active site. That's one of the key words. Anytime lock and key theory or lock and key hypothesis is mentioned. So I'm going to say starch or substrate, either is fine. I'm going to go with starch. And I'm going to say binds to the active site. And just to be clear what I'm referring to, I'm going to also add here of amylase. So that's going to be my first point, and as you can see, it's a 3 marker, so I'm going to need to come up with 2 more of these in my explanation. So my next point that I'm going to refer to is what happens, or why why this starch is able to bind to the actual active site. So something we have to mention is this idea of complementarity. So the active site is designed in a way so it's complementary to the shape of the substrate, or vice versa. So what that means is that they're a similar shape, as in if this here was an enzyme, I'm going to stick a little E there, your substrate could be this. And this is able to like dock into that bit there because it's got a complementary shape. So that's going to be the next bit that I'm going to write down. So I'm going to go and say the shape of the active site is complementary to substrate. Ooh. So what that means is that they can fit together, but simply saying that they're complementary is enough to get the mark. And what that means when this binding takes place, because the last part of this question is going to be your answer, sorry, is going to be referring to how it breaks down starch. So after the binding of your substrate and enzyme, after that happens due to the complementarity, because this is breaking down starch, you can say that what's happening is bonds are being broken between molecules within starch to produce smaller molecules. Or you can say chemical reactions occur to produce smaller molecules, because you need to refer to, though, that you're forming smaller molecules because you're breaking down starch, which means you're breaking it into things which are smaller than itself. So here what I'm going to go with is the chemical reactions explanation, but I'm also going to say chemical reactions breaking bonds occur. So I'm going to go chemical reactions breaking bonds occur to form smaller molecules. Okay, so here you can see how we first analyzed what we need to look at. So you've got an explain, you need to refer to amylase and starch and their breakdown, and answer this in terms of the lock and key hypothesis. And the first 2 points, where your substrate, in this case starch, binds to the active site due to complementarity, that is the lock and key hypothesis, which is of this idea. The idea that part of the substrate is the same shape, or the shape isn't the same, but shapes are complementary, allowing them to fit into each other. A bit like a hand and a glove, you could say. So the next part of the question is going to be: a student investigated the effects of temperature on the activity of amylase. Okay, the figure below shows the apparatus used. So we've got a test tube, 5 centimeters cubed of starch solution, that's in this tube, beaker, so big beaker which everything's put into, water at 5 degrees, and then a 1 centimeter cube of starch solution. So there might be some flipping of pages here. So this method was used, and now we've got the method. So set up the apparatus as shown in the figure above on the other page for us. After 5 minutes, sorry, let me just make sure you can see that fine. Yeah. After 5 minutes, pour the starch solution into the amylase solution and mix. Remove 1 drop of the starch amylase mixture and place into a spotting tile. Immediately add 2 drops of iodine solution to the starch amylase mixture on the spotting tile. So we also need to think, what's the point of adding iodine here? Record the color of the iodine solution added in the starch amylase mixture, and then repeat steps 2 to 5 every minute until the iodine solution stays yellow-brown. So what does this possibly mean? And then repeat steps 1 to 6 using water at different temperatures. So, hopefully you've all processed that, or at least some of it. If you're watching this back as a recording, feel free to just pause and think that through. I am going to go ahead with at least thinking about this first question. So here we've got name 2 control variables the student used in the investigation. So here they don't want us to come up with some control variables. They want us to name some that the student used. So the first thing that we need to think about is what is a control variable. So that's going to be something that stays the same between each iteration of your experiments to make sure that your results are comparable, which means that, like, for example, if you used 2 liters of water to grow one plant versus 10 liters of water to grow the same plant just in a different environment, those results might not be as comparable because the amount of water used was not the same every time. So, if we now take a look at this example, what can we think of here? So 2 control variables, and we can have a look at our diagram that they gave us. So I guess 2 things that stick out to me is, I'll come back to the water in a second, but you've got fixed volumes of starch and amylase. So fixed volumes means that every time you redid this in terms of repeat steps 1 to 6, that included setting up the apparatus as shown in that figure, that means that that every time they will have 1 centimeter cubed of amylase and 5 centimeters cubed of starch. So those are probably the 2 biggest things that would stick out to me. So you could say here, volume of starch, and here you could say volume of amylase. So some other things that you could refer to is that here you said immediately add 2 drops of iodine. So that also means that every time is only going to add 2 drops of iodine. And so that also could be considered a control variable. Another thing that could be noted is that they placed only 1 drop of the starch amylase mixture onto the spotting tile. Ooh, underlined the wrong bit. So there. I would also say if you're a fan of highlighters, use them in the exam rather than having me underline stuff, because that's the way I'm used to doing it. If you prefer using highlighters, definitely like underline, circle, actually highlight, and like pretend that was a highlight. Apologize if we need to reread that. But do so because it definitely helps you remember what the key features and key points that you need to refer to are. So next we have, why did the student leave the starch solution and amylase solution for 5 minutes before mixing them? So this is referring to step 2. So after 5 minutes, then pour the starch solution into the amylase solution and mix. So why was were the two tubes left in the water bath for 5 minutes before adding one solution into the other? So to look at this question and to sort of understand where they're coming from with it is if we look at our very last step here. So repeat steps 1 to 6 using water at different temperatures. So what that means is that temperature is going to be one of our independent variables, but either way it's something we want to accurately know and control. And leaving our tubes in for 5 minutes means that they get to heat up or cool down to the same temperature as what is on in the water itself. So it just means that both tubes will be of the same temperature. So that's the answer that I'm going to go with. So I'm going to say to allow both solutions to reach the same temperature, and to be specific you can say temperature of water. And that's the bit that I really want to focus on there, because it's not that they reached the same temperature just by chance, is they both reached the same temperature as the water in the beaker, and they've told us here 5 degrees, so you could also technically write allow both solutions to reach 5 degrees Celsius or something along those lines, and you would also still get the mark. So we're going to try and have a quick look at these last two questions here and then see what we can do. So the table below shows results of the investigation. Temperature in Celsius in one column, time taken until iodine solution stays yellow-brown in the other column. So at 5 degrees, it did not turn yellow-brown at all, apparently. And at 20 degrees, it happened after 5 minutes, at 35, at 2, at 57, at 65 degrees, after 14, at 80 degrees, it did not become yellow-brown, same as 5. Okay. So, our first question is going to be, what conclusion can be made from the effects of temperature on amylase activity between 20 and 65 degrees Celsius? So this is going to be between this bit here. So the first thing that we can give reference to is what is the general trend we see. So here time decreases, but then after this point, so 35 to 65, the time taken increases. So that's quite literally what I'm going to write. So I'm going to say as temperature increases, so does activity for T, and I'm gonna say up to 35 degrees C after which you can be very cheeky and add another line on the bottom in these. What does it say after which? Activity decreases. If you've got more than just literally 2 words to write for a question, flip to the back of your paper. There'll be some extra paper there. If it's not in the back of your paper, ask one of your exam invigilators for some extra paper. All you need to do in the end is just sign it, and they can take it, add it to your paper, and just make sure you write the question number associated, and just say MC printout or handout or something of the sort, or extra paper would be best, just to make sure. So let me just check if we have any questions. No questions for now. Okie dokie. So let me know if there are any questions on anything so far. I am going to just try and tackle at least some of this last question. So here we've got explain the results at 5 degrees and at 80 degrees C. So this is a 5 marker, so we're going to have to put some thought into this one. And it asks us to use the table above to explain the results at 5 and at 80. So this solution, iodine solution, remaining yellow-brown at 5 degrees, tells us what? So remaining yellow-brown means that the iodine test has come back negative, so there is no starch. Oh, sorry, no, it's come back positive, other way around. So the starch is still there, sorry, because it's not turned yellow-brown. And what that means is because originally we had starch, which is amylose, and amylase, which breaks down starch, at 5 degrees it was too cold for any activity of amylase. So they did not have any or enough kinetic energy to be able to make collisions, and thus no reaction. So that's going to be the first bit that I'm going to write about, and I'm going to say at 5 degrees C, molecules have low kinetic energy. You might be tempted to write Ke if you're doing physics, but unfortunately this is not physics and it's not one of the accepted abbreviations, so don't write Ke in here. You have to write the full thing out, kinetic energy, but in physics, depending on your exam board, you can write Ke. So low kinetic energy means that there are fewer collisions. Few collisions, and what we can say is that collisions between enzymes and substrates. So what that means is that we don't get any activity, and that explains the results at 5 degrees. It hasn't said describe, so you don't really need to go what that means. But if you wanted to, just to cover all bases, you could say, so starch is present. So now you can add at 80 degrees that there is the opposite thing. Well, no, it's the same thing happening, but for opposite reasons. So what I'm going to say is that 80 degrees is that what happens is the temperature's gone too high, and we know here that this here is a loss of activity, and loss of activity at high temperatures is going to be due to denaturation. So at 80 degrees, amylase is denatured. And what this means when denaturation happens is that your substrate can no longer fit into the active site and can no longer bind into the active site. So that means that starch is unable to bind to the active site. And I guess one thing that you could add to this point here is that, just to make sure this is clear what you mean by starch is present, so you could say starch is present, so there is no yellow-brown color, or you can say it remains blue-black. I'm just going to say, so no yellow-brown color. Right, that will be it from me today. I'm trying over by a few minutes, but I'm sure that is fine. Thank you everyone for coming. I hope this has been useful. I hope we've gotten at least something out of this in terms of how we can look at questions and think about applying our knowledge to questions and not just regurgitating everything we know, which is a lot of the time what you think you need to do, but you need to be very careful about what the examiner is looking for to get the marks that get you the grades you want. But this will be it from me today. We should hopefully be back next week for another one of these, just half an hour on another topic. If there's something in specific you want us to look at, please leave us a comment or shoot us a message somewhere on Instagram would be great. And we can try and have a look at one of those topics for you. So yeah, like I said, that's it from me. Hopefully this has been useful. Best of luck with the rest of revision, and I will see you there.